Connaissance.
Recherche.
Sciences.
Des sciences mathématiques.
Théorie.
Physique.
Philosophie.
Conférence.
Archéologie. Nature.
It's really a great pleasure to be here in this glorious institution. And today we had a taste of what it is to be invited to a meeting in France. I was at Harvard 2 weeks ago, and we had lunch with postdocs with a little turkey sandwich. And today here we had this glorious feast with salmon and cheese and wine. And so feel free to invite me anytime. So my talk today will be on AIDS vaccines, and particularly I'll try to discuss the angle of natural HIV infections, which I think has the two keywords that my colleague Lucia Mori just discussed a few minutes ago, the learning from nature and the concept of being non-conventional. So what I will do, I wasn't sure how many people in this room are familiar with the field of AIDS vaccinology, so I decided to add a couple of slides to summarize where the field of AIDS vaccinology stands today, and then I will go on into discussing what I think can be learned from nature in this field, and I'll like to share with you a few ideas on how to potentially move the field forward by exploiting these ideas in the design of AIDS vaccines. So I think everybody agrees that the current status of AIDS research is the half full, half empty glass. We can certainly rejoice, the number of incredible discoveries that have been done over the past 20-25 years, but there's some major stumbling blocks that have been still hampering our progress, particularly our inability to prevent infection with a vaccine and our inability to eradicate infection. And I personally think that a lot of this has to do with the fact that we don't completely understand the pathogenesis of HIV and AIDS. So the main premise of any discussion of AIDS vaccine is that we have to train the immune system to do something that doesn't do in nature. In other words, there is no natural immunity against HIV infection. So it's really a different type of challenge when we want to make a vaccine for HIV as opposed to a vaccine for other infectious diseases like smallpox or polio or measles and so on and so forth. And because of the peculiarities of HIV infection and AIDS, I often like to show this lovely painting from René Magritte. It's called The Empire of Light, and it's a beautiful painting. And you probably all have the experience of going to a museum and having something that really stays with you for a long time. So this painting stayed with me because I looked at it for about 10 minutes, and I was thinking how beautiful it is. And then somebody told me, do you realize that this is a surrealistic painting? It's something that doesn't make any sense logically. It's beautiful, but doesn't make sense. And finally, I realized that the top half of this painting is a broad daylight, and the bottom half of this painting is night. So you can't have day and night at the same time. So what the painting is showing is impossible, but it's still beautiful. So I think sometimes, AIDS vaccinology has been the same way. People came up with beautiful theories that just had the little problem of being impossible. And since we can't just look at them, but we actually have to use them, that approach, the sort of surrealistic approach to AIDS vaccinology didn't work. So one example of a potentially, surrealistic approach to AIDS vaccinology was the STEP trial. So this was a trial, it was sponsored by the NIH and Merck, where the idea was to elicit CTL responses against HIV antigens by making the immune system see them in the context of a vector. In this particular case, it was a non-replication compound, human adenovirus serological type 5, expressing 3 different genes of gene products of HIV. And the result of this study that was made public in the fall of 2007 is that when you look at the vaccinated people versus placebo, in the overall population of patients that were vaccinated, there were 49 versus 33 cases. And particularly if you look at the individuals with high titers against the vector, the human adenovirus 5, there were 21 cases in the vaccinated versus 9 cases in the placebo. And this was deemed to be significant in the original analysis. There have been, interestingly, a number of post hoc analyses of this trial that all tend to dismiss this result as some kind of a quirk, some kind of a statistical, not particularly important statistical strange result. I still look at this and I think that maybe there's some message there, and it's just not just a statistical quirk. There was also another trial that was completed about a year ago, actually a little bit less than a year ago, and the results were, I think, first presented in June 2009. And this was a trial that was hailed with considerable skepticism by the community of AIDS scientists. So this is the typical trial that had a lot of trouble starting. As a matter of fact, in 2004, Science published this letter, and those are the authors of that letter. And you recognize a lot of famous names in the field of AIDS vaccinology. And where basically the idea was we should not spend the money, waste the money doing this trial because there is no rationale. And the study went on anyway and came up with this big surprise that in fact 30% of people were protected from infection. And as a matter of fact, this is probably the single most encouraging news in the field of AIDS vaccinology since this field started. So the reason I mention this is because it's not because I have anything against these people. If I had been in 2004, I probably would have signed that letter anyway, because that was exactly my feeling. So it's not like I want to criticize them. I'm just saying that really this field has been marred by a certain dose of surrealism, in the sense that things that we thought were going to be very useful, potentially very promising, like the STEP trial, turned out to be either ineffective or even potentially dangerous, and things that we thought were not going to do anything actually end up showing some kind of protection. So what is the problem? Why is this happening? I think one of the reasons this is happening is because in the field of AIDS vaccinology, there's been a problem, something that I see as a major problem, and maybe others don't necessarily agree with this, but I think a major problem has been the idea that all we had to do is try to repeat some vaccination approaches that have been found to be useful, to be effective against other viruses or other pathogens. And I think that approach, it may be, I think, interesting in theory, but in practice collides with the fact that HIV has some unique properties from the biological, immunological point of view, so that make its interaction with the immune system substantially different than that of other viruses like polio, smallpox, or measles, or varicella, for which we have effective vaccines. And particularly, this is the key point that I will emphasize over and over in my presentation, is that HIV is a virus that infects activated CD4 T cells. HIV is a virus that infects activated CD4 T cells. This is something that's been known for over 20 years. And for some reason has really had a hard time filtering into the field of AIDS vaccinology. So what does it mean? It means that every time we try to make an immune response against the virus, there is the intrinsic risk of creating more targets for the virus, therefore creating worsening the risk of disease transmission of infection or even disease progression. And I think that, I still think that the results of both the STEP trial and the TIE trial can be really seen under the light of this simple consideration, and everything makes much more sense. So in other words, what is the challenge here? The challenge is that we have a very strong evidence that a number of effector mechanisms of the immune system, particularly CTL and antibodies, are capable of potently suppressing virus replication. On the other hand, we have some aspects of the immune responses, and particularly the activation of CD4 helper cells, that make life easier for the virus. So here comes the balancing act. So the idea is that we might have to figure out a way to have strong antiviral immune responses without giving too many target cells to the virus. And I think this is, if I were, in the position of deciding policies for where the resources should be put in basic AIDS vaccine research, this would be, to me, priority number one. As a matter of fact, I'm absolutely convinced that a vaccine that is perhaps not very potent in inducing antibodies or CTLs, but is very good at keeping the activated CD4 cells away from mucosal tissues, might end up being more effective than a vaccine that induces very strong CTL responses, but also packs up the mucosa with target cells. And I think that's exactly what happened with the STEP and the TIDE trial. So I'm now going to switch gear and talk about the natural HIV infection, and I will go back to this issue of the target cell, and I'll try to explain that that's exactly what nature is telling us, that maybe the best way to fight with SIV and HIV infection is to work on the target cell side, not just on the adaptive and innate immune responses against the virus. So natural SIV infections are important for a number of reasons. First, are important because that's where HIV-1 and HIV-2 come from. Thanks to the great work of Beatrice Hahn, now we know that HIV-1 is the result of cross-species zoonotic transmission of SIV-CPZ that infects chimpanzees in Africa. And HIV-2 is the result of cross-species zoonotic transmission of SIV-SMM that infects the Pseudomangabes. And the SIV of Pseudomangabes is also of interest because it's the direct progenitor of SIV-MAC, which is the virus that has been used for many years as the model of AIDS vaccines and AIDS pathogenesis in Asian macaques. And Asian macaques are monkeys that are non-naturally infected with the virus. And if they get the virus, they develop AIDS. They're like humans. All these African monkeys here, and I only show 8 in this slide, but there are 40 species known so far. Each one has their own individual species-specific SIVs. So far, none of them has been shown to develop any sign of AIDS. Instead, chimpanzees that are more recently infected are now known to have a disease that is less severe than HIV-1, but certainly it's a real phenomenon of pathogenicity as opposed to the non-pathogenic infections of the African monkeys. So just to give you an idea of what this field of comparative AIDS research, meaning AIDS viruses infecting different monkey species, I will just show quickly on this plot by virus replication and pathogenicity where the different models stand. So I'll start with HIV-1. We all know HIV-1 is a pathogenic infection which with a variable level of viral load, but usually is a pathogenic infection with uncontrolled replication. There's a small subset of lucky infected individuals that we refer to as the elite controller on long-term non-progressors that have low viral load and do not experience AIDS. Then there is HIV-2 infection, which is something in between HIV-1 and the long-term non-progressor in terms of pathogenicity. Infection of macaques, the experimental model, usually results in high levels of virus replication and a course of disease progression that is even accelerated compared to HIV-infected humans. There are also some lucky macaques that have a phenotype of elite controllers, and then we have chimpanzees that, as I said before, have a sort of a moderately pathogenic infection, sort of comparable to HIV-2 infection in humans. So when you look at all these models, it's very clear that there is a relationship between pathogenesis and viral load. More virus, you have more pathogenesis, and vice versa. When you add to the picture the natural infection of those African monkeys, and here I'm plotting the Pseudomangabes, and this is an African green monkey, the main difference is that we have, during the chronic infection of adults, high virus replication and no pathogenesis. Now, in this slide here that I'm rapidly filling, because I don't want to spend too much time here, I'm listing a series of characteristics of the pathogenic infection of humans and macaques in two columns here, and the non-pathogenic infection of the natural host here. And some of those things I already mentioned, so you don't have to spend too much time, like the absence of AIDS, the high viral load. There is a preservation overall of the CD4 count in peripheral blood. Overall, there is a maintenance of mucosal immune function, even if there is some certain degree of loss of mucosal CD4 cells. What's really interesting is that the virus appears to be equally cytopathic in all species, and it's been measured both in vitro and in vivo with a series of experimental systems. But there are some striking differences between these models, and those are the three differences that I will discuss today. The first is the immune activation. During the chronic phase of infection, both SIV infection of macaques and HIV infection of humans are characterized by high levels of immune activation, and this is something that is not present in the natural SIV hosts. Also, another feature that has been recently discovered in natural hosts is that there is much less virus replication in CD4 central memory cells as opposed to effector memory cells when compared to the pathogenic models. And finally, there is much less vertical transmission from mother to offspring in these models of infection than in the pathogenic models. And this is the current model in the physiopathology of these infections, with the two key elements underlying the lack of disease progression being the resolution of immune activation, the fact that the chronic phase of infection is associated with low immune activation, and the fact that the virus has low levels of replication in central memory CD4 cells. And we think, a lot of people in the field think that these two components sort of feed off each other in a sort of a virtuous cycle that keeps the animals away from disruption of the CD4 pool and eventually disease progression. So what I'm going to do in the next few minutes is discussing three aspects of the infection. I will discuss the antiviral immunity, what we know about the way these monkeys develop immune responses against the virus, particularly the adaptive immune responses. Then I'll discuss the role of low immune activation and target cell restriction, those 2 points that I mentioned here, and how they may apply to an AIDS vaccine concept. And then finally, I will go quickly over some data on the vertical transmission of these infections from mother to child. So this is a paper that was done in our lab by my former graduate student, Rick Dunham. What we did here was we measured the CD8 T-cell responses against SIV peptides using conventional intracellular cytokine staining assay in a number of SIV-infected pseudomangabes. And in our paper, we compared them with SIV-infected macaques. But in this slide, I'm showing historical comparison with data that were generated by my good friend Mike Betts in a study that was published in Journal of Virology a few years ago and confirmed in several other studies. The point I'm trying to make is that when you measure the magnitude of CD8 T-cell responses in humans with HIV infection and in pseudomangabes with SIV infection, you have this very paradoxical finding that you have more responses, stronger responses in the model where there is disease and lower responses in the model when there is no disease. And I know that a lot of conventional vaccinologists don't like these results, but that's a fact of nature. This finding has been reproduced in other labs and also in African green monkeys. So it's a feature of natural infections that they're associated with lower T-cell responses to the virus. And these lower T-cell responses do not put the animal at any risk of AIDS. It's interesting that recently Cindy Dardine and in collaboration with my lab published a paper where we looked at the neutralizing antibody titers in the pseudomangabes. That was the first study of antibody neutralization of autologous envelope proteins. And what we found was really interesting because we did not find any sign of neutralization of autologous virus in these animals. So in humans, for those who are not in the field, in humans there is a phenomenon that is quite interesting, that if you take today's virus in the average HIV-infected patient, that today serum of the average HIV infected patient, that serum will neutralize the virus that was present in the patient like a year ago. But if you take that serum and you try with that serum to neutralize the virus that is today in that patient, there will be no neutralization. In other words, there is this continuous chase of the virus by the B cells and the humoral immune responses, which is always a step or maybe multiple steps behind the virus and never results into an effective antibody-mediated clearing of the virus. Now, in these animals, that phenomenon doesn't occur, and the animals don't even bother to make neutralizing antibodies against the old virus, which we think is an interesting phenomenon. Most recently, just a couple of months ago, this paper by a great colleague of mine, Jörn Schmitz from Harvard, showed that in the AGM, you can actually deplete CD8 T cells and B cells during the acute infection. So to me, this is the most radical intervention you can make, short of shooting the animals, to abrogate their adaptive immune responses. And even in the presence of in an infection, it was done in absence of CD8, so in absence of CTLs and in absence of B cells. Still, these animals were able to live with virus replication that wasn't significantly changed by these interventions. And the animals survived this treatment. If you do the same treatment in macaques, you get accelerated disease, and they, in fact, die in a few weeks. So what is the point here that I'm trying to make? Well, the point I'm trying to make, going back to what we heard before from Lucia, is the concept of learning from nature. So what is nature trying to tell us? And again, we are free to listen or not listen to nature. I think usually listening to nature is a good idea, but the point that nature is trying to make is that a deal with HIV infection, with AIDS virus infection, was reached in these animals that have been infected for thousands and thousands of years, not by creating a very, very strong adaptive immune response that would pretty much kill the virus or block virus replication, but rather by mechanisms that are independent of strong immune responses. And in fact, these immune responses, if anything, are lower than what you see in the pathogenic model. So what is the specific feature of these animals? And here comes the concept of low immune activation. So there's a number of studies that have been published by many groups over the past 6, 7, 8 years that all show very clearly that the chronic phase of infection in the natural host, in these African monkeys that don't develop AIDS, is associated with low immune activation. What do I mean by low immune activation? I mean a number of things. For instance, production of pro-inflammatory cytokines. I mean number of cells expressing activation markers or proliferation markers. I mean apoptosis, bystander apoptosis. I mean disruption of the lymph node structure, disruption of the bone marrow function, disruption of the thymic function. So the chronic immune activation is something that is associated with pathogenic HIV and SIV infection, and there's a number of reviews in the field that I can address you to if you're interested. But the point is that what is the thought is that this chronic immune activation ends up basically destroying the ability of the immune system to respond to newly encountered pathogens. And therefore, in this context, the ability of tuning down the immune activation is something that helps the animals dealing with other pathogens, other potential infection, and therefore protects them from developing AIDS. So how do we translate that into AIDS vaccine concepts? And this is something that sort of bizarrely has not got into the field. So the field of AIDS vaccinology has been often, I think this slide probably is one of the most commonly shown and discussed slide in the entire field of AIDS vaccinology. The concept is that you have a vaccine that will not prevent infection, but will prevent disease progression by lowering viral load and therefore making the infected individual living longer and not experiencing rapid disease progression. So this is the model. So here you have, in red, you have the vaccinated individuals that have CTL against the virus. They will have a smaller peak of viral load, and they will have a lower level of viral loads, and this lower level of viral loads will protect them both from transmission, but especially from disease progression. Now, viral load is a very strong marker of disease progression, but actually immune activation is as strong as viral load. If it could be, in fact, even stronger than viral load. So one possibility is to have the anti-inflammatory vaccine paradigm, and I showed this slide at Keystone, in the Keystone meeting on AIDS vaccine a couple of weeks ago, and I got a lot of people coming to me and say, oh, that's interesting, and I was thinking, this is something that the field has known for many years. So the idea is that if you plot the level of immune activation, if you have an intervention that blocks the level of immune activation along the lines of what Patrice Debray was discussing before in terms of vaccination against pathogenesis instead of vaccination against transmission, you could have an infection with lower immune activation early on, lower immune activation in the chronic phase of infection. And if this low immune activation goes below the pathogenesis threshold, you could have these people actually live a potentially normal life. The target cell restriction is the other big lesson that we got from nature, from these natural hosts. So the point is that this is an observation that we made a few years ago in collaboration with Ivona Pandre at the University of Tulane. So what we observed is that there is much less CCR5 expression on CD4 T cells of natural hosts when compared to the non-natural host in both peripheral blood and mucosal tissues. And actually, when you look at mucosal tissues, it's a quite dramatic phenomenon. Those are CD4 cells of rhesus macaques and baboons. Those are non-natural hosts. And you see this large fraction of cells expressing CCR5 in the large intestine. If you go into the same cells of sooty mangabeys and African green monkeys, you see this dramatic decline. And those are uninfected animals. So this has nothing to do with the infection. This is just the way these animals are. They have learned to express much less CCR5 on their CD4 cells. So for a long time, we have been sort of puzzled by this observation. And we thought, what does it mean? These animals have high viral load, so therefore they must produce enough virus, and they seem to transmit the virus efficiently in the wild. So what does it mean to have this low CCR5 on CD4 cells? And I think we're getting to the bottom of it. There are a couple of areas where this is important. One is the protection of the central memory CD4 cells from SIV infection. And this is what I discussed before when I was talking about pathogenesis. So the fact that central memory CD4 cells are protected, we think it's a key element to induce low immune activation, and low immune activation is a key element to induce lower infection of central memory CD4 cells. And you have to remember that the central memory CD4 cells are probably the essential cell to maintain a sufficient pool of CD4 cells despite virus replication. In other words, what matters is not how much virus you have in the body, but what cells are producing the virus. And if the cells producing the virus and killed by the virus are cells that are less important to the overall homeostasis of the immune system, you can deal with that without experiencing major trouble. The other point is that the low CCR5 expression of CD4 cells may protect natural hosts from vertical transmission. And this is a vastly understudied area of AIDS research in these animals, but we're starting, compiling a few results that I'll share with you in the next few minutes, and I think they might have some serious implication in terms of AIDS vaccine design. So when we look at mother-to-child transmission, I think you all know that this is a major tragedy in the developing countries, and when you pull together all type of mother-to-child transmission of HIV, in utero, intrapartum, and breastfeeding related, you can come up with numbers in the range of 30 to 50%. In rhesus macaques, there is one study looking specifically at breastfeeding transmission, where 14 female macaques were infected at the time of breastfeeding, and 10 of 14 promptly transmitted the virus to their offspring. We recently conducted a study in sooty mangabeys, and we looked at 162 animals born to SIV-infected mothers. They were tested for antibodies against the virus both during the first and second year of life. 147, so 90, close to 91% of these animals were clearly not infected vertically because they never became seropositive, or they have an interval of seronegativity when they cleared the antibodies coming from cross-placental transmission from the mother, and then they became positive again at early stage in life. We got 15 animals, 9%, that have consecutive positive tests, serology positive tests in the first and second year of life. And these animals may or may not have been infected vertically, but when we looked at viral load, interestingly, we were only able to amplify virus in 7 out of 15 animals, which means approximately 4% of the animals. And 6 out of these 7 animals have viral load less than 500 copies per ml. Now, the average viral load of Pseudomangabes, adult Pseudomangabes, is over 100,000 copies per ml. So these young animals have 2 to 3 logs less viral load than the adults. So they were very rarely infected, only about 4 or 5% of cases. And when they were infected, they had very, very low viral load. And similar data were also generated in mandrills and African green monkeys. In particular, in African green monkeys, there's a study that I want to show you that was done by Vanessa Hirsch and Jason Branchley at the NIH. And they kindly allow me to show these slides, still unpublished, but I think very interesting. So what we did, we took an infant African green monkeys and we infected with SIV-AGM high levels. So the idea was to see, is it possible that the infants of these animals are susceptible to the infection, so that the reason they're trying to protect them from mother to child transmission is to avoid disease. So and again, this is one case, so you have to take it with a grain of salt, but I think it's really interesting, the results. So the animal seroconverted, so developed antibodies. This was an animal born to a non-infected mother, so these are antibodies from the baby. Seroconverted made the classical antibodies against the GAG and envelope proteins of SIV. But this animal had basically a flat line of viral load. In other words, we could not detect viral load in the periphery. Now, this virus that we used is perfectly replication competent. In fact, was used to infect adult monkeys. And you see the usual range of set point viral load in African green monkeys, around 100,000 copies per ml of plasma. This baby monkey had no depletion of CD4 cells. And on top of that, when we did a CD8 lymphocyte depletion, there was no reactivation of the virus. In other words, the viral load was low not because the CD8 cells were controlling the virus. You probably know that in some models of non-pathogenicity in adult monkeys, you remove the CD8 cells and viral load picks up again. In this particular case, it had nothing to do with the role of CD8 cells. So at this point, we don't know exactly how this baby was able to become infected, seroconvert, and therefore mount adaptive responses to the virus, but still keep viral load very, very low. But one possibility, we think an intriguing possibility, is that this animal had very, very low levels of CD4 positive cells expressing CCR5. And in fact, we know from other studies, for instance, one that we recently published in mandrills, looking at expression of CCR5 in CD4 cells of newborns, that newborns of these animals have extremely rare frequency of CD4 positive, CCR5 positive cells. So again, what is the lesson of nature? Well, the lesson of nature is that this rarity of maternal to child transmission of the virus is most likely not related to strong adaptive immune responses in the baby, but rather by a particularly low level of target cells. So we're going back to the target cell issue. We protect by lowering the target cells, not by going to war in terms of adaptive responses against the virus. And one idea that we and others are entertaining is that perhaps the evolutionary pressure to protect newborns from infection is what created the adaptive immunological premises to then make the infection non-pathogenic in the adults. So I want to finish with a couple of conclusion slides and a couple of thoughts for reflection and for potentially new ideas. So what is nature telling us? I think nature is telling us something quite unconventional and unexpected, and perhaps even, savor for some of us in the field. But that's what nature is telling us. It's telling us that, first of all, that AIDS is not just a consequence of having the virus in the body. That AIDS is a much more complex and complicated effect of the interaction between lentiviruses and the host immune system. And in this interaction, not all immune responses against the virus are good. And I'm talking about good as delaying disease progression or delaying transmission. Sometimes these responses can be bad. Two key features of the natural infection, the low immune activation and the lower level of target cells, may be important concepts to explore in the field of AIDS vaccinology. And particularly the idea that babies in this species can be protected from infection, from vertical transmission of the infection, by having very low level of target cells. I think it's a very fascinating idea that really should make us think differently about what is the type of immune response that we want to generate with an AIDS vaccine. So these are this is my last slide, those are a few thoughts for AIDS vaccinology. For instance, and those are things that if I were the one making the policies of the NIH or the NRS or other, funding agency, I would say put the money on these ideas rather than chase, the 20,000 spots, ELI spots, or the 50,000 ELI spots, or whatever, a million ELI spots that you want to get. So, for instance, is it possible to uncouple CD4 T cell activation and CD4 T cell homing to mucosal tissues from CCR5 expression? So, is it absolutely written in the stone of nature that every time the CD4 cells go into mucosal tissues, they have to be CCR5 positive? Because if you think about it, if we can get the CD4 responses in mucosal tissues elicited by a vaccine made by cells that don't express CCR5, I'm sure that's going to really tip the balance very strongly towards protective responses versus non-protective responses, even if maybe the number of CTLs there or the titer of antibodies there is not particularly high. But just the lack of these cells there can make the environment extremely unfavorable for the virus. Also, can we uncouple mucosal HIV-specific CD8 T cells and B cell activation from mucosal CD4 T cell activation? To what extent, when we generate an antiviral response in mucosal tissues, to what extent we actually need the activated CD4 cells to be there? And I'm not aware of studies showing what is really needed. I mean, we all agree that we need to have CD8 T cells there against the virus. We all agree that we need to have antibodies, possibly neutralizing antibodies, maybe even non-neutralizing antibodies, maybe ADCC-inducing antibodies. But to what extent having these cells in the mucosal tissues is dependent on the actual presence in there of activated CD4 cells? Also, to what extent we need CD4 cells to provide this type of help? And there is some intriguing data in the monkey model suggesting that in some cases, non-CD4 cells can also provide quite effective help, for instance, in the African green monkey model. And the other issue that I think it's important point for discussion is, can we consider vaccines that will result in breakthrough infection that will develop in the context of minimal immune activation and potentially minimal disease progression? Is that an idea worth exploring? I mean, remember that this field for years thought that a vaccine that would lower viral load of 1 or 2 logs or 1.5 logs would be the bingo. And I mean, if you look at the studies of HIV prognosis, 1 or 1.5 log of less viremia is the equivalent of having maybe half of the immune activation that you have otherwise. In other words, you could have the same exact result of lowering viral load by lowering immune activation. So I'll finish by referring to a paper that we recently published in Nature Medicine where we discussed some of these concepts and there are also other concepts that I didn't have time to discuss due to time limitation. And I want to finish by acknowledging a large number of collaborators in my lab at Emory, other institutions, Particularly, I want to acknowledge Jason Branchley and Vanessa Hirsch for sharing the data on the baby African green monkeys, Ivona Pandrea and Tracy Meeker for sharing some of the data on the young mandrills. And all my money comes from the NIH and some Concerned Parrots for AIDS Research, W.W. Smith Foundation, and the Penn Center for AIDS Research. Thank you for your attention.